Okay, now we are going to go over the structures of the inner ear. To begin with, we have the semicircular canals. These help us with balance and detecting rotational movements. These can be a little tricky to figure out which one is which one because all three semicircular canals have their own name. In this model, they are placed in their anatomical position so we can name them fairly easily. To begin with, we have our lateral semicircular canal. That is right here. It points laterally, so that is why it is called our lateral semicircular canal. We then have our superior semicircular canal. Here, again, it points superiorly, giving it its name. Then we have our posterior semicircular canal. Again, this is the most posterior canal, giving it its name. In this model, it is conveniently placed in anatomical position, but that may not be how it's tested. So if we pick it up and move it around, it can be a little bit trickier to name them. So again, we have our lateral semicircular canal, our superior semicircular canal, and our posterior semicircular canal seen here. I would recommend in lab getting a model and moving it around to see if you can continue to name the semicircular canals. At the base of each semicircular canal, we have our ampullae, which are these bulges here. These contain sensory cells that contribute to our sense of balance. Moving away from our semicircular canals, we now have our cochlea. We are going to take it out and move the model so we can see it a little bit better. Here we have the cochlea. It looks like a snail shell or a cinnamon roll if you're hungry. And going off of it, we can see the vestibular-cochlear nerve here. If we cut into the cochlea and we open it up, we can see these chambers shown here. We are going to zoom in and look at a larger model of these chambers. Okay, now that we have zoomed in really far into the cochlea, we are going to look at this cross-section here. So first we have the Scala vestibuli. That is this whole space here. This is the upper chamber that contains a fluid called perilymph. Separating the Scala vestibuli and the cochlear duct is the vestibular membrane. So vestibular membrane. This, again, is the cochlear duct, also known as the scala media. The fluid that fills the cochlear duct is called endolymph. Farther down, we have the scala tympani. This is the lower chamber of the cochlea, which is also filled with perilymph. So we have the scala vestibuli and the scala tympani, and then the cochlear duct. Let's take a look at this structure called the organ of Corti. Here we have the basilar membrane. This membrane will vibrate at different frequencies. On top of the basilar membrane, we have hair cells. These are the hair cells. As the basilar membrane vibrates, the hair cells will push against the tectorial membrane. This is the tectorial membrane. As the hair cells push against the tectorial membrane, mechanically gated ion channels will open and allow the endolymph of the cochlear duct to flow into the hair cell, which will depolarize the hair cell. This can then be turned into an action potential that is sent to the brain via the vestibular cochlear nerve.